Okay, so note that these are blue, and they come in a little holder. Okay, so we're going to put that. We're going to just we can use our pipette carefully, and you don't want to get. You want to get as little or none of that white goo. The goal is to do this without getting the white goo. All right, so we have to suck that out of there. In fact, you might do well to just pour it in. I think that's what I'll do. I'll just pour it in. Pour it in, and that's it. Okay, that's good. All right, one. Okay, and we'll do the other one. So you only need to do one of these. I'm doing two because I need more plasmid for a separate project. But, right. So now, after we pour this in here, okay, I have the supernatant. I poured it onto the column. I'm going to go now and put it back in the centrifuge. So, and I'm going to centrifuge it for 30 to 60 seconds, and that's it. So once for 30 to 60 seconds. Basically, what we've done here is we've lysed the cells, and what we're pelleting here is the chromosomal DNA and the some of the cell debris, like parts of the membranes and that kind of thing. And we're taking the supernatant, and we are going to be now. Basically, it's designed so that you can pellet all of the large molecules, and what's going to be left, the plasmid DNA, is small compared to the chromosomal DNA. So that one stays in the supernatant. And so when we put it on the column, we are going to purify out everything except the plasmid. So the plasmid is now attached to the column, which is what we want. So when we spun it through, all of this liquid down here has other cell debris in it. It doesn't have the plasmid. So the little column is designed to adsorb the plasmid DNA. But we've got the chromosomal DNA out in the precipitation step. So that's how this system works. Okay? So discard. We're going to discard this. I'm just going to discard it. And the next thing is we're going to wash the Chi-Prep spin column by adding 0.75 milliliters. So we'll set our pipette to 0.75. There it is. It reads 0.75 from top down. And now I'm going to wash it with buffer PE. And note that the buffer PE says on the top of it, ethanol added. Yes, and there's a check in the box. That means somebody put the ethanol into this buffer. Don't use any buffer PE that doesn't have ethanol in it, because it won't work. You will be very sorry you did that. Okay, so we're going to put this in here, and this is to wash everything out of the column that we don't want. All right? So this step keeps the plasmid on the little white column part, but we're going to wash off all of the other cell debris that is still trapped in it. A nice clean plasmid trapped in that column. And the same thing would work. There's slightly different buffers that we're going to use to purify out the PCR product. So it depends on the size of the DNA, which kit you use. That's how the buffers have been designed to maximize the particular, your choice of DNA size onto the column. So that's what this step is for. So the next step we're going to do is going to tell us to, we did this wash step with the buffer PE, okay, and then it's going to tell us to remove the residual wash buffer and do it now one more time. Okay? So you're going to have to remove the residual and then spin it again. Okay, you will find in here buffer EB. This is the buffer that you want to use for the elution. And this is really important for your steps. In order for us to get a high enough concentration of the plasmid in your product, the instructions tell you you can elute with 50 microliters of buffer EB. But that's too much, okay? You're going to want to use only 30 microliters, okay? So we're going to elute with 30 microliters. In all of the steps that we do, we will elute with 30 microliters. So how are we going to measure 30 microliters? We're going to get a pipette that reads either P200 or P100. Here's one that reads P100. The challenge with P100 is to make sure that you find a tip that fits. And we will have two clean tubes. And so just take the little blue thing out and put it into a clean Eppendorf tube, just like that. And I will do the same. Get rid of this bottom part. And then I'm going to take the other one out of the other tube and put it in a clean Eppendorf tube. Okay, there we go. All right. There we go. We are ready. And now I'm going to take my pipette, adjust this to 30. And we're going to load in 30 microliters of buffer